All right, guys. Big deal here. Gene regulation. Your cells, as humans, have 46 chromosomes in the nucleus, 2 sets of 23, your diploid, plus 1 extra chromosome found where? In the mitochondria. You get that mitochondrial chromosome from which of your parents? Your mom. All your organelles, that's come from mom. What you do not want to happen is you don't want every gene on all of those chromosomes to be working full speed everywhere. Your genes get regulated. In your liver cells, there's a certain set of genes that are working. In your brain cells, there's a whole different set of genes that are working. In your pancreas, there's a different set of genes that are working. That's what we call selective gene expression. That only certain genes are turned on in certain places. Because we can do that, that lets our cells differentiate, become different. That's why a liver cell is different than a stomach cell, which is different than a brain cell, which is different than a pancreas cell. Because the DNA in all those cells I just described is exactly the same. It's just different pieces of the DNA are turned on in those different cells. Now, in terms of regulating our genes, the question up here is why regulate? Let's say that JP just drank one of those big AMP energy drinks. What just happened to JP's blood sugar? It just shot up because they're loaded with sugar, right? As soon as that happens, his body needs to deal with that because high blood sugar is a problem, right? So what genes in his body need to get turned on real quick to deal with that? Genes for making insulin. But do the genes for making insulin all over his body get turned on? They only get turned on in what organ? The pancreas. The pancreas starts making insulin, chugging it out into the blood. The insulin goes out into the blood. It goes mostly to the liver and the adipose cells. And it says, hey liver, my blood sugar is too high. Here's our blood sugar, these little markers floating around in the blood. There's too much of this in our blood. The message to the liver is to grab that glucose out of the blood and do what to it? Put it together. Put it together and make what polysaccharide that my chain of markers here represents. Insulin causes your body to make what polysaccharide out of glucose? Glycogen. Not glucagon. Glycogen. And the liver and the muscles store that glucose. But they're taking the glucose out of the blood, therefore lowering the blood sugar. What's the message to the adipose cells? Hey fat, too much glucose floating around. What do the fat cells do? They take sugar, convert sugar to fat, therefore doing what to blood sugar levels? Lowering. Alright, let's say JP now. Back to JP. Let's say JP says, hey, I'm just love fat. Because look at him. I mean, he is fat, right? JP's really fat. So JP just says, hey, I'm not going to eat for a couple of days. Just takes off a couple of days. What happens to his blood sugar levels? They start to drop. And his body says, whoa, can't let that happen. We don't maybe need JP to be hypoglycemic because that'll kill him. So what genes get turned on? Genes for making what get turned on when his blood sugar starts to drop? Glucagon. Glucagon starts being released from cells in the pancreas. It goes out to the liver, out to the adipose, and it says, hey liver, you got this glycogen stored up in you. We need it. So what does it do? What does glucagon cause to happen to the glycogen? It causes it to be broken apart. And what gets released when it's broken apart? Glucose molecules, which bring the blood sugar back up. The message to the adipose cells. Say adipose, you got a lot of fat in there. Do what to it? Convert it back from fat into sugar. Raise my blood sugar levels. So that's glucagon and insulin. Let's say that JP, imagine this, has been a really bad student today. He's been getting on my nerves, which, sometimes he can do. And I just decided I had enough. So I just grab it up and start popping it, okay? A physical thing, okay? Some days, I might want to do that, really, okay? Occasionally. What genes better get turned on in JP?
Fight or flight.
Good genes for making adrenaline, right? But do adrenaline genes get picked up all over JP's body? Only where? In his adrenal gland. Okay? Adrenaline back up here, thank you. And his adrenaline starts, his adrenal gland starts making adrenaline, which will prepare him for fight, which probably wouldn't be a good idea. Flight might be a good idea, right? Right. Flight might be the best response, okay? So, regulating your genes help you to respond to your environment. Chemical, physical, and even also hormonal kinds of things. We'll talk about gene regulatory proteins more in a minute. Let's first of all talk about bacteria. When scientists first started trying to figure out how genes get turned on and off, they said, hey, let's start with something that we have a chance of figuring out. Let's start with bacteria. Because they only have how many chromosomes? One. In what shape?
Circle.
Circle, loop. Not necessarily a circle, but it's looped, right? And what do they not have that we have? Attached to their DNA. Histone proteins that will school, they don't have those. They also don't have introns, which we'll talk more about in a few minutes. So they started with bacteria because bacteria DNA is pretty simple. And they said, hey, let's figure out how bacteria turn on and off their genes. And what they found is bacteria have these structures called operons. Important point. Prokaryotes have operons. You, as a eukaryote, do not. Let's look at a picture to talk about what these operons do. That's all we're getting today. That's how we regulate. The most important part of an operon is this right here, the structural genes. Those are the genes that we're trying to control. We're either trying to turn them on or off. It's a set of genes that we're trying to control, structural genes. But the operon is all these things. The operon is the genes plus the pieces that help to control these genes, that help to regulate them. Key word today is regulate. So it's the genes and the structures that help bacteria to regulate these genes. The operon basically has 5 parts. The structural genes, the promoter, promote. That ought to clue you in. That's like the starting spot, right? Promote means to help get started. I told you last time we talked about this that the most important thing that I was going to tell you about that time was this statement. What 2 things have to be happening to a gene for that gene to be on and working? Transcription followed by translation. DNA to mRNA, that's transcription. mRNA to protein, that's translation. tRNA helps, right? But it's mRNA to protein, that's translation. So here's the deal. Transcription has to happen first. What's the enzyme that makes transcription happen? What enzyme makes RNA from DNA?
Polymerase.
Which polymerase? Yeah, but what's the thing that goes in front of the word polymerase? RNA polymerase 2, right? This is the binding site for RNA polymerase. That's what the promoter is. It says, hey, beginning of a gene, start transcription here. That's the promoter, promoting transcription. It's the binding site for RNA polymerase 2. Now here's the other thing. This operator is in between the promoter and the structural genes. The operator is where a repressor may or may not be. Repress means what? Stop. Stop. Prevent. So if a repressor is bound to this operator, RNA polymerase would be over here. The genes it's trying to transcribe are over here. That repressor is like a roadblock. If the repressor is on the operator, what can we say about this set of genes? They are off. If there's no repressor, guess what?
It's on.
It's on. So that's really what you need to know about that. Now, somewhere else in the DNA, maybe not in this straight line, but somewhere in that bacterial chromosome, there's a gene called a regulatory gene or regulator gene that codes for the repressor, because the repressor is protein. So you've got promoter, operator, structural genes, Regulatory genes and repressors. Those are the main parts. Now there's two kinds of operonds that turn out to be important. One kind is called an inducible operond. What's the word induce mean? Make happen. Make sure you understand the statement I'm about to make. Inducible operonds are normally operonds that are off in bacteria. They're normally not working. Inducible means you can start. It's normally off, but under the right conditions, it can be turned on. That's an inducible operand. Something that the bacteria normally doesn't need to do, but in a pinch, it may need to. So it's an operand that's normally off, but can be turned on when it needs to be. The example that you need to know of an inducible operand is called the lac operand. Lac standing for what? Lactase. Lactase is the set of enzymes, proteins, that digest what? Lactose. Bacteria, most bacteria normally don't make a whole lot of lactase because they're normally not exposed to like milk. Let's say that you're a bacteria living right here on the palm of my hand. And I'm walking around, there's no milk on my hand, and so you're not making lactase because that'll be wasteful, right? Don't make it if you don't need it. But then maybe at lunch, I decided instead of eating a nutritious lunch, I'm going to take chocolate chip cookies and I'm going to dip them in glasses of nice cold milk. And when I do that, some milk splashes up on my hand, you can wash off right into the cup of milk. So now you're a bacteria, you're surrounded by what? Lactose. And it's free food. But right now you can't eat it because your genes for lactase are what? Are off. So you need to turn them on. You need some selective gene expression. So here's how it turns on. Notice right now the repressor is bound to the operator, which means RNA polymerase can't make it over here. So there's no transcription going on. This gene's off. But now down here, now there's lactose. It's all around you. You're a bacteria. There's lactose everywhere. That lactose actually comes in and attaches to the repressor. And when it does, what happens to the repressor? It came off. And when the repressor came on, what happened to the operand? It's on. See, the repressor came off. The operand is now on. Did I say it backwards? Repressor comes off, operand comes on. RNA polymerase slides across this stretch of DNA like a train on a track. DNA used to make RNA. That's what process? DNA to RNA. Transcription. RNA to proteins. Translation. Remember, enzymes are a kind of protein, right? So this would be lactase. Because lactose helps to turn on the operand, turn off the repressor, lactose is sometimes called an inducer. So the actual substrate helps to turn on the genes that are ultimately going to do what to it? Break it down. That's an inducible operand. You should know that the lac operand is an example of an inducible operand. Now the other kind of operand is repressible. Repressible operands are normally on, but can be, what's repress mean? Turned off. The example that you're going to hear about for repressible operands is called the trip operand. Now, you need to know that the trip, that abbreviation, stands for tryptophan, which is an amino acid. It's an amino acid that bacteria have to have, but they usually don't get it where? It's an amino acid bacteria have to have, but they usually don't get it where? From their diet. So therefore they have to what? They have to make it themselves. The trip operand is a set of structural genes that code for enzymes that make tryptophan for the bacteria. So the operand doesn't code for tryptophan. Because what do genes code for? Genes code for proteins, not for amino acids, right? They code for chains of amino acids. So these structural genes code for some enzymes that will make tryptophan for the bacteria. The bacteria normally doesn't have tryptophan, so these genes are normally on making. You can see that. Structural DNA, mRNA, enzymes. Transcription, translation. But let's say all of a sudden, this bacteria, you're back on my palm again here. Not getting any tryptophan, so you have to make your own. It's a bacteria. But all of a sudden, it's Thanksgiving, and I start carving turkey. And you know, and I have to butt my hand up against the turkey, and you stick to the turkey. You're the little bacteria. What is turkey loaded with? Tryptophan. There's lots of tryptophan in it.
What is tryptophan?
It's amino acid. Okay? All of a sudden, these bacteria are surrounded by tryptophan. Free. It's there for the taking. Does it make sense to keep making it if it's all around you? No. So this operon is going to get turned off. Tryptophan binds to the repressor and turns the repressor on. If the repressor is on, it's now going to stick to the operator and therefore turn off what? The operon. So tryptophan acts as what we call a co-repressor. Tryptophan turns on the repressor, which turns off the operon. Now let's say the bacteria eats up all the tryptophan in the turkey. What's then going to happen? It's going to come loose. It's going to turn back on. That's that negative feedback stuff, right? Feedback inhibition.
So is tryptophan, if the repressor turns off the opposite?
Yeah, when tryptophan binds to the repressor, it activates the repressor. The repressor sticks to the operator. Operon is off.
Up there at the top, if the repressor, then the thing's working?
Yeah, then the repressor is inactive, but the operon is active. Making mRNA, making proteins. Not just a second, just wait a minute. All right, now, we'll come back to operons in a minute. But first of all, let's talk about eukaryotes a little bit. All that operon business that we just mentioned, all that operon business that we just talked about is only in bacteria. We are way more complicated. When, I'm waiting on everybody to focus here. When scientists first started doing the Human Genome Project, which they were trying to figure out what each gene in a person does, they thought they were going to find thousands of genes in people. Did they end up finding more or less genes than they thought they were going to find?
Less.
Way less. We don't have that many more genes than some way less complicated organisms. But here's what scientists figured out. For a long time, they thought that a lot of our DNA was junk DNA, that it had no function. It didn't seem to code for what? For anything. It didn't seem to code for protons. And they called that proteins. They call that DNA segments that don't seem to be coding for anything introns. But here's what they found out. In eukaryotes, remember, we do transcription to make RNA. Let's say this was a piece of RNA. All right, piece of mRNA. The first piece of mRNA that's made in eukaryotes is called the primary what? Transcript, right? Primary transcript. Before that can get translated in eukaryotes, what has to be done to it? In eukaryotes, there's transcription, and there's something else that happens before translation. What has to happen to that mRNA? It has to be processed, which means what has to be taken out? There shouldn't be
The introns,
the intron sequences, right? So let's say that this goes to be processed. What are the enzymes that do the processing? Take out the introns, called spliceosomes. Well, let's say that whatever's going on in your body right now, these three bases get treated as introns. So they get cut out, and then what happens to the exons? They get spliced back together, right? Splice means connected. They get spliced back together. And then we get a protein from that. That gets translated.
Splice means that the other one spliceosomes take stuff away.
Well, they cut things out and then put the other people together.
So they do both.
They do both. That comes from like old-fashioned film, like film on a reel. If something happened to that film, and maybe it got whited up, a couple frames did, you would cut out the whited up part and just tape the other pieces back together, splice them back together. That's where that comes from. But let's say next time around something different is going on in your body. Your body needs to make a different protein. So instead of this CCC getting treated as an intron, maybe this gets treated as an intron. And now what do you get after translation? You get a completely different way. Protein. Different RNA, different protein. One gene is not just coding for one way. Protein. In eukaryotes, in humans, one gene can code for lots of proteins because of what we call alternative DNA splicing or alternative processing. Make sure you know that in people, in eukaryotes in general, there's transcription, then processing, then translation. Not only are introns removed during processing, what also happens during processing? What gets put on the 5' side of the RNA? Cap. And that's there to keep it from getting broken down by your body. And what gets put on the 3' side of the RNA?
Tail.
The tail, called the poly what? Poly A tail. It's a bunch of adenines get stuck on the end. That's processing. Now, in terms of us regulating our DNA as eukaryotes, our DNA gets regulated on 3 levels: transcription level, processing level, translation level. We regulate our DNA on 3 different levels: transcription, processing, translation. So let's talk about the transcription level here first. Let's first of all talk about how steroid hormones work. Steroids, what kind of chemicals? Lipids. Where are the receptors for steroid hormones at? Inside the cell, because they go straight through the cell membrane, right? Important point. You're going to see this in the study session Saturday too. Steroid hormones go in and they bind to receptors inside the cell. That hormone receptor complex is now like a key. It goes straight to where? Straight into the nucleus. Let's say, it's going to make a bad analogy to it. It goes straight into the nucleus, attaches directly to the DNA. What did it just do? Turned on a certain gene. So steroid hormones, the way they're going to act, they're going to cause what kind of reactions? Since they go straight to a gene and turn it on. Fast. They're going to act very fast and very dramatic, right? You're going to get a big response because their receptors are in the cell. They go straight to a gene, turn it on. You get fast response, big response. A protein hormone, like insulin, for example. Insulin's made of protein. Where's the receptors for that at? Where's the receptors for insulin at? It's a protein hormone. Outside the cell. Pay attention. A protein hormone, first of all, has to bind a receptor on the outside of the cell, cause that receptor to change shape. We call that the changing of the shape of the receptor, conformational change. That turns on a protein in the cell, which turns on another protein, which turns on another protein, which turns on another protein. It's that whole signal transduction pathway, right? Until eventually, we end up getting what? What's the last part of cell signal? A response. But since it has to go from receptor on the outside through a whole long pathway of signal transduction to response, how does its response compare to a steroid hormone response? Way slower and way less dramatic. Does that make sense? So you should know, here's what you ought to know. Steroid hormone receptors are in the cell. Steroid hormones go directly to the DNA, turn on specific genes. You get a fast response and a big response. Protein hormones, receptors on the outside. They bind to a receptor. The receptor is passed on through a long signal transduction pathway. It's kind of slow, and the response is not very dramatic. It's not very big. Does that make sense? All right. So that's an example of what happens at a transcription level. Testosterone, for example. Testosterone goes into the cell, binds to the receptor on the inside of the cell. Testosterone will then travel into the nucleus, and it will turn on what I like to call the manly genes. Okay? Notice how my voice dropped there? The manly genes. Turns on the manly genes. Okay? And that you get a very quick response and a big response when that happens. Back at the beginning of the year, we talked about an issue where this went bad. We talked about what's called androgen insensitivity. What is an androgen? What is an androgen? What does andro mean? Like android? Man. Android's like a man. And androgen is a male sex hormone, like testosterone. These people are insensitive to androgens. These are the guys who are born, and the doctor says, you have a beautiful baby daughter. Because on the outside, they look like girls.
Can they not tell by the parts?
No. On the outside parts look like girl parts. Internally, though, internally they have testes. No ovaries, no uterus. Internally, they're testes. Those testes, yeah, I'm about to tell you a thing here. These testes are chugging out lots of testosterone. But what's messed up? The receptors for testosterone are made the wrong way. So the testosterone's there, it's just their body doesn't know what to do with it. Okay? So the basic sex in humans is women. Without testosterone or without working receptors for testosterone, you come out looking like a girl. There's a big controversy going on right now in the Olympics. There is a, quote, woman with this disorder who wants to compete against who?
The guys.
She wants to compete against the women.
That's cheating.
Okay? Here's the deal. There are different degrees of this syndrome. Some of the women with this syndrome basically are completely insensitive to testosterone. So it's like it's not there. So really, that would be fair, woman against one of those people. But there's also situations where these women are slightly sensitive to testosterone, which means since they have testosterone in their bodies, it's like they're taking what? Steroids. It's like they're taking testosterone, and they're going to become more muscular, bigger, stronger, faster than a normal woman.
They have both parts.
No, just female parts on the outside. Here's the deal. This woman, if you look her up, just when you go to the computer, Google androgen insensitivity and Olympics, and it'll be the first link you find. This woman is very buff. It's a black woman from Africa. Very big, strong, fast.
They got some
No, just because they don't have female parts.
What do they have?
Internally. They're female on the outside, but male on the inside. No ovaries. So it's like a male
So
female.
they don't have their menstrual cycle.
No, there's no ovaries that do that. No uterus. But here's the deal.
So they're men.
Except on the outside.
So they're it's.
So here's the thing. This woman is a little bit bigger and stronger than most women. And she wants to compete against the other women. A lot of the other women say, whoa, that's not fair. But the decision of the Olympic committee was to what? They're going to let her compete against the other women. Which is really, I don't think fair at all. But that's the way it came down. These people, though, does she think like a man? Think like a woman?
Both.
Hard to say. Okay? Now, if she was completely insensitive to testosterone, she'd think like a woman, right? Because even though it's there, the testosterone's not changing the architecture of her brain. Testosterone changes our guy's brain. I know that you ladies cannot believe that us guys think differently than you do. I know you can't believe that. But we do. Okay, we really do. Our brains are different than your brains.
Is it true that women see more birds? Like dust?
They notice it more?
Yeah.
Probably. I mean, us guys, we don't pay attention to details sometimes, right? You ladies are a lot more complicated than we are, okay? If two guys get into an argument, they punch each other and then shake hands, right? It's over. Not the case with you ladies, okay? Not the case. Not the case. But back to what I'm saying. This is an example, this whole testosterone thing, of how we control our genes by regulating transcription. Here, join me, if testosterone is not there, or if your receptors for testosterone are not working, the genes, the manly genes that I'm calling them, they never get transcribed. So it's like they're off, right? So that's an example of a The transcription level. Now, another thing, and I want to show you a little article here. Go ahead.
Brittany Griner had that.
I don't know. But she's a pretty manly looking person.
Yes, it is. She
I don't
talks
know.
like a man.
She's huge.
Bit fast.
She's
Yes, that.
girls basketball player, dunking in games. So, I mean, it's basically nothing up top, right?
It's not even.
Um,
It's not even fun for her. She's like, she's just grabbing.
it doesn't really look fair. All right, I've got an article here that I want you to read real quick. Let me get it pulled up. I'm gonna pause the recording for a minute. All right, guys. Today, tackling the cardiovascular system, the heart, and you've seen on these system tests, there's always a diagram. There'll be a diagram. You got to know the parts of a heart. Hey, you guys working on outlines? Let's put that stuff up. Pay attention to what we're doing here. First of all, the heart sits in like this. So the point, notice it's got a little point on it called an apex. Apex, like the apex of a building is like the point, like at a church, right? The point always points left. You need to know that. The point points left. Apex points towards your left side. On the outside, there's several things you need to notice. First of all, on the surface of the heart, you've got these blood vessels, these arteries, called coronary arteries. We've talked about those before. Hugely important, coronary arteries. Even though your heart pumps all your blood, can it use most of what it's pumping? It cannot, okay? What actually feeds the heart, gives it oxygen, gives it nutrients, helps it get rid of waste? The blood going through these guys. The coronary arteries. Try that again. Having technical difficulties this morning.
So they carry the wavelengths?
Yeah, up here the aorta, one of the first branches of the aorta is to these coronary arteries. These carry blood and oxygen to the heart for it to use for itself. If those get clogged up, what do you got? That's where heart attacks happen. Okay? So when they start talking about doing bypasses and stuff, triple bypass, they're talking about those. Those are the arteries that get clogged up that cause a heart attack. So the coronary arteries take the blood to the heart muscle. The cardiac veins bring it back into regular circulation. So these are a pair. Coronary arteries, cardiac veins, they work together. Coronary arteries to the heart, veins back into regular circulation. So you need to know what coronary arteries are. If they get clogged up, you have a heart attack. All right, some other vessels that you need to be very familiar with. The biggest artery in your body, right up front here. Right here in the middle, notice it's kind of shaped like a horseshoe up at the top. That's the aorta. It's like the interstate through your body for what kind of blood? Oxygenated blood. Okay? It's the main interstate. All the other roads, all the other arteries branch off the aorta. And then the opposite of that are these two vessels. The superior and inferior vena cava. Superior just means what? Above the heart in this case, and inferior means below the heart. But those are the biggest what in your body? The biggest veins. They're like the interstate for what kind of blood? Deoxygenated.
Hold on. Where's the aorta?
The aorta is a huge one right here, huge red one. I don't know that it is. It's just like it says A or T.
And then all blood goes to the aorta. Or goes
All the oxygenated blood starts out in the aorta, and then it branches out from there into different arteries. The superior vena cava and the inferior vena cava. Those are the major veins in your body. It's the interstate, we'll call it the interstate for deoxygenated blood. General rule, arteries always carry blood which way? Away from the heart. And usually it's what kind of blood? Oxygenated. Veins always carry blood which way? To the heart, and it's typically what kind of blood? Deoxygenated. Here's the exception to that rule. Here's the exception. This vessel right here. These are called the pulmonary arteries. And notice they're blue. You don't usually see arteries colored blue. What does pulmonary mean? Speak up, you know. It means lungs. Pulmonary means lungs. Okay? This artery carries blood to the lungs. What kind of blood needs to go to the lungs? Deoxygenated. Okay? Where it will get oxygenated, and then it will come back to the heart through the other exception to our rule that we just talked about. These little guys over here and over here, notice they're called pulmonary veins, but they're colored red because they're carrying blood coming from where to where? From the lungs back to the heart. So anytime you see that word pulmonary, realize it means lungs. Like CPR stands for cardio pulmonary resuscitation. You're bringing back their heart and their lungs. Cardiopulmonary resuscitation, CPR. So those are the major things in the picture you need to be familiar with. This picture. Now this is kind of an outside view. In a minute we'll look at an inside view of the heart. You're going to have to know both. All right, just some general things. The circulatory system or the cardiovascular system. General functions you already know. It transports nutrients. Those nutrients get into the blood mostly from what digestive organ? Small intestine. Okay? Small intestine is the main digestive organ. It's where absorption of nutrients takes place. Also dissolved gases. Oxygen, CO2 are carried in the blood. Oxygen is mostly carried by what protein in your blood? Hemoglobin. Okay? And then also hormones. We know hormones travel through your blood as well. We know that as far as the blood vessels go, in humans, in mammals, we have a closed circulatory system, which means the blood cells never leave the blood vessels. It's a closed loop. The liquid parts of your blood can leave. Gases and stuff can leave. But the cells themselves, they can't unless you cut a vessel. These are the vessels that make up that closed loop. I'm going to start with arteries. So you've got arteries coming away from the heart. Those arteries will start to branch out. They'll start to split. And they split into smaller vessels called arterioles. Those continue to split into smaller vessels called capillaries. Capillaries are really small. Usually the walls of a capillary are only one cell thick. You want them to be thin because what needs to happen at a capillary? That's where all the good stuff that the blood does happens. Oxygen out, CO2 in. Nutrients in, waste out. It's where gas exchange happens, nutrient exchange happens. In the kidney, it's where the glomerulus is at, right? It's where all that filtration stuff happens. So the capillaries need to be really, really tiny, really, really thin, because diffusion and osmosis and all that stuff has to happen there. But after that's done, they start merging back together. Think of it like an interstate. The capillaries start to merge back together and make bigger roads. Those are called venules. They're starting to head back toward where? Back toward the heart, so we're looping back now. The venules merge together to make bigger roadways called veins. And then eventually the veins all merge back together to make the interstate. And what do we say the interstate of the veins is? Just from that diagram, what was the interstate of the veining system? Superior and inferior vena cava. So they all merge back and go to the heart. So it's a closed loop. Like we said, we have a closed system. Our blood cells do not leave our blood vessels, but nutrients, water, oxygen, those things get exchanged at these capillary beds that are traveled out throughout our body. Some organisms have an open system, and that means they have a heart, or something like a heart, and a couple of main vessels, and it just sprays out. It bathes their organs. A lot of times their blood is not actually called blood. Sometimes you'll see it called hemo milk. I'm not sure why they make the distinction, but sometimes they don't call their blood blood. Hemo means what, by the way? should have learned about this in chemistry. It means iron. We know that hemo has to do with hemoglobin, right? Iron-containing protein. So in terms of open systems, when we talked about all our animals, we talked about how the arthropods, like insects and crustaceans, had an open system. Mollusks, what are mollusks? Clams, shellfish, octopus, squid, snails, slugs, that kind of thing. The open system is not nearly as efficient as our system, right?
You know when the blood just like works out.
Yeah, they just put their organs through a blood bath, basically. All right, let's talk about the differences between arteries and veins. We already talked about the differences in direction. Arteries, which way? Away from the heart. Veins, to the heart. But their structure is different too. Arteries, the walls of the arteries tend to be really elastic, really stretchy. They tend to be really thick and strong. And we also already mentioned they tend to carry oxygenated blood. So remember, it's coming from the heart. The heart just pumps, which means the blood coming through those arteries has a lot of what behind it? A lot of pressure. If the artery walls weren't thick and strong and stretchy, what would happen? They would burst. And what do we call that in the body when a blood vessel bursts? Swells and bursts. An aneurysm. You do not want that to happen, especially to your aorta. All right, your veins, on the other hand, by the time the blood gets to your veins, it's already been around the loop, right? Which means what's happened to most of the pressure? It's kind of went way down. The blood's moving a whole lot slower. It's not moving very fast. It's not moving with a lot of pressure behind it. So veins, the walls of veins tend to be a whole lot more thin, not very stretchy, low pressure. They are typically carrying deoxygenated blood. And something unique to veins, they have little valves in there. So let's say blood's coming back up your leg. It's not coming very hard. So sometimes, especially if you stood up, what might be the reaction of your blood? If you were laying down, all of a sudden you stood up, what might the blood try to do in a vein? Go backward. You want your blood to start flowing backward. That's a bad idea, right? So it's got little valves in there that once the blood comes through, they close, and they won't let it go backward. The valves are to keep the blood going forward, to keep it from backflowing. Veins are the only vessels in your body that have those. Make sure you know that veins have valves to keep the blood from flowing backwards, because that blood has very little pressure behind it to keep it going forward. Who usually has problems with their blood circulation? What group of people? A lot of times it's people who don't move around a whole lot. Because your veins, one of the things that helps the blood flow through your veins is moving. Because when you're moving, what are your muscles doing to your veins? They're squeezing on, pushing on, okay? And what's that creating inside those veins? Some pressure. It's pushing the blood, okay? People on airplanes, like long distance flights, like the one I was on from transatlantic, that's hours. And if you haven't flown in a plane lately, the seats are smaller than these desks, right? I mean, your leg room. And so your legs are all bent up. And if you just sit there for 12 hours, what's happening to some blood in your legs, possibly? It starts to pool up. And it might form what? Plaques. That's something you don't want, right? Because those clots can break loose and move from your legs into your lungs, into your heart, into your brain, where they can get stuck and kill you, literally. There are people who die every year because they've been on a plane for a long time and they just sat in their seats and the blood clotted and it killed them. When you're on those long plane rides, you should get up, walk occasionally, go to the bathroom, make a loop around the plane, stretch out, do a little jogging or whatever. Just get up and move your legs, get that blood flowing. Yeah. And that causes flash.
No, I'm not a golf coach, but she didn't really like that. It's like above my arm, and then I moved up here the next day, and like a fever the next day, and then she got checked out. She was freaked out and something like that.
What did they have to do to it?
I think they just like had to give her a shot or something.
To break the clot? Yeah, that's kind of dangerous.
Yeah.
Blood clots can kill you in a hurry. All right, let's talk about the structure of a blood vessel. Now, make sure you're paying attention to what I'm telling you here. The innermost layer of a blood vessel, the inner wall, is called the endothelium. You know endo means inside, right? So the inner wall is the endothelium. It's made of a kind of tissue called simple squamous. Simple squamous is like epithelium. It's like skin-like tissue. It's very thin. Simple means it's one layer thick. Squamous means it's flat. So it's one layer of flat cells. This actually makes up the tube, though. It makes up the lining of the inside of your blood vessel. Every kind of blood vessel has this endothelium. Capillaries, arteries, arterioles, veins, and venules. They all have it. It's nice and smooth inside. If you had a rough surface inside your blood vessels, what happens when you have rough surfaces to blood? They clot to it, right? That's why when you put gauze on a cut, right? Because it's all rough, it's got a lot of surface area, and the clots form when they come into contact with rough surfaces. You do not want the inside of your blood vessels to be rough. It's not a good thing. They want to be very smooth. That's another reason why people tend to have heart attacks, because if you eat a lot of fat, what happens to some of that fat in your coronary arteries? It deposits on the walls of those coronary arteries and makes them wet. Makes them rough. What starts to form on those rough walls? Blood clots, and that causes heart attacks. So the innermost layer of a blood vessel, endothelium, very smooth, very thin, provides a smooth surface area for the blood to go through. All right, the next thing you have in your blood vessel is smooth muscle, a layer of smooth muscle. We talked a little bit about smooth muscle before. Ricky doesn't have much of it. Smooth muscle is not that kind of muscle. You know, this kind of muscle right here, you know, that's skeletal muscle. That is voluntarily controlled, right? Consciously controlled. Smooth muscle is the muscle that makes up like your organs. Can you consciously control your organs? No, it's unconscious, involuntary muscle. Same here. But those muscles in your arteries and arterioles and in your veins, notice that venules and capillaries are not listed here because they don't have this layer. That muscle is for peristalsis. Well, that's a good word to know. Anybody know what peristalsis is? The muscles in those artery walls contract. And as it contracts here, what does it do to the blood that's in there? It pushes it. And then what happens to the next section? It contracts, and then the next section, and then the next section, and then the next. So it's like squeezing toothpaste from the bottom up. Pushes the blood through. That process of peristalsis is also real important. Where else in your body? Not just the esophagus, the whole digestive system, right? Your whole digestive system is made of muscle, and as it contracts in a rhythmic pattern, it pushes the food through the digestive system. Peristalsis. Important word to know, peristalsis. Just like a wave-like contraction of muscles. And in this case, it's pushing blood through the blood vessels. In your digestive system, it's pushing food from your esophagus to your stomach, from your stomach to where? What comes next after stomach? Small intestine. From small intestine to large intestine. From large intestine to toilet, right? That's the typical way it goes. Behind the smooth muscle layer is a layer of connective tissues that hold it all together. Connective tissues, what protein should you think about when you hear that word connective tissues? Very strong, holds your body together.
Collagen.
Say again. Collagen. Okay? Collagen. When you hear the word connective tissues, I want you to think collagen. Collagen is a protein that holds you together. It's like the glue and the rope that binds your body together. So there'll be a lot of collagen in this. Strong. Keeps your arteries from bursting. Keeps you from having aneurysms. Notice that's found in all of the blood vessels except the capillaries. And then we also mentioned that the valves are found only in the veins. And those keep the blood from going backwards. Keep it going in one direction. Unidirectional flow. One direction. Unidirectional. Blood going backwards is a major problem. So those are the different kinds of layers in your blood vessels. Endothelium, nice and smooth and thin. Smooth muscle to push the blood through the blood vessels. Connective tissues to hold it all together. And by the way, I posted this PowerPoint and the outline last night, so you can get these and use them. This is showing you how the arterioles connect up to the capillaries, which connect up to the venules. And it's showing you some differences between the structures. These are really thin and small, the capillaries are. The arterioles are much thicker, stronger. Notice the layer of muscle here is way bigger than the layer of muscle here. The thickness of the wall is bigger. But now let's look at this. This is showing you a capillary bed. Remember, arteriole, capillary, capillary to venule, venule to vein, back to vein, back to vein, back to the heart, heart to an artery, and then it starts all over again. It's a closed circle. In this capillary bed, that's where all the diffusion and osmosis and gas exchange and nutrient exchange and waste exchange and all that good stuff happens. Stuff comes out of the blood there. Stuff except the cells themselves. What's all this green stuff? Notice these are the lymph vessels. A lot of people don't understand the lymph vessels. You have two main sets of tubes in your body. You have your blood vessels, and you have your lymphatic vessels. Their main job is to recollect liquids that leak out of the blood. So a lot of stuff comes out of the blood vessels. They recollect that liquid, and eventually up here in your chest, do what with it? Put it back in the blood. But before they put it back in, what do they do to that liquid? They clean it, they filter it. That's what the function of what part of the lymph system is. The lymph what? What you've all heard of. Lymph nodes. What's found in lymph nodes. There you go. Lots of white blood cells. And that's where they filter the blood and check it for infections and such. This is showing you in a grasshopper, an insect, an arthropod, how his blood system is not closed. Heart, vessels, and then nothing. They basically put their organs in a hemolymph bath. We'll use that word instead of a blood bath. All right, an annelid. Phylum Annelida are like earthworms and what else? These are segmented worms, so these are like earthworms and leeches. There's also a lot of ocean-dwelling worms and marine worms that are segmented like this. These guys have a closed system. How many of you dissected an earthworm in regular biology class? Most of you should have. Most of what you see in an earthworm are two things. Most of it in the inside is its digestive system. It goes from mouth to pharynx to Kroppen gizzard and to intestines. And its intestines are like this long, okay? Why are his intestines so long? More surface area for what? Absorption of nutrients from the digestive system. Yours is the same way, right? If they were to slice me open and start ripping out intestines, they'd go to that corner over there. They're really long. The small intestine is really the large intestine, right? Small intestine is bigger than the large intestine in terms of length, way bigger. Because it's about absorption. But the circulatory system is the other thing that you can see pretty well inside an earthworm. It's got a couple of main vessels, and it's got 4 pairs of these blood vessels that pump. So it's like it has 8 hearts to some extent. And they pump the blood in a closed loop. Very much like ours. Whoops, get a little ahead of myself there. Something else we want to talk about, and then we'll do a little lab activity about later, is blood pressure. You need to know about blood pressure. Typically, when we measure the blood pressure, we're measuring the pressure in an artery, not a vein. We do it right up here, and usually don't do it here, but there's a way to measure blood pressure here in the throat area. What's the main artery in your throat? Don't say jugular, it's the jugular vein, right? It's the artery here in your throat. You've all heard of this artery, I know. It's called the carotid. Carotid artery. But most of the time we measure it in your arm, right? They put those blood pressure cuffs on and they measure the pressure in your arm. Any idea what the name of that artery is? The one in your arm. Say again? It's the brachial artery, right? So usually those are the two that we measure the pressure in. You can measure it in any artery, but those are the two we usually measure it in. We're measuring two things when we measure blood pressure. When your heart contracts. When your heart is contracting, is the pressure going to be higher or lower? If your heart just did this, pressure going to be higher or lower in the arteries? Higher, because it pushes, right? When the heart contracts, the word for the contraction of the heart is systole. And that's a word you need to know. That means the ventricles of the heart, the big chambers, are contracting, which means the blood pressure should rise, correct? Systole. This is when the heart contracts. It's a word for the contraction of the heart. Brianna, join us, please. The relaxation of your heart is called diastole. So contraction followed by relaxation. Pressure should go up. Pressure should go down. So when you measure your blood pressure, when you go to the doctor and they tell you your blood pressure, they tell you two numbers, right? Something over something. The number at the top is called the systolic pressure. That's the pressure when the ventricles of your heart are contracting. It should be the highest part of the pressure. It should be somewhere between you ladies, it's going to be a little bit lower in you, right? Because your heart's not as big and strong as this big old guy, right? So your pressure's going to be a little bit less. Should be 100 and something though. Somewhere between like 110 and 130. Pretty good range for systolic pressure. The bottom number is called the diastolic pressure. And remember, that's the pressure when your heart is relaxing. It should be lower, somewhere around 70. It might be a little bit lower, a little bit higher. But a kind of an average, normal, healthy blood pressure is around 120 over 70. Yeah, you should know that. An average, normal, healthy blood pressure is somewhere around 120 systolic, 70 diastolic. Let's think about some things that can affect your blood pressure. We talked about aldosterone, a hormone made by your adrenal gland. What did aldosterone cause you to keep in your body and keep out of your urine? It caused you to retain sodium and therefore retain what always follows sodium around, water. If you have more water in your body, more water in your blood vessels, you're going to have more pressure in your blood vessels, right? So that can make your blood pressure go up. Too much sodium can give you high blood pressure. And by the way, what is the scientific name for high blood pressure? It's a word you should know. High blood pressure. What's the more sciencey medical name for it? Hypertension. All right, if your blood vessels were to dilate, vasodilation, what should happen to your blood pressure if most of your blood vessels did that? It should get less, right? Because now you got bigger pipes, same amount of fluid, pressure should drop, right? On the other hand, if your blood vessels constrict, they get more narrow, your blood pressure should go up. That make sense? So vasodilation should cause blood pressure to decrease, Vasoconstriction should cause it to increase. Vaso just means what? Vessel, blood vessel. Vaso. Some people say vaso, some vaso. Potato, tomato, tomato. Vaso. I don't care what you say. Either one. All right, let's talk a little bit about the actual makeup of the heart. It's mostly made of a type of tissue called cardiac muscle. Cardiac muscle is kind of unique because it's self-stimulating. What does that mean, self-stimulating? What does it not need that other muscles do to move? Nerves. All the other muscles, the smooth muscle, the skeletal muscle, a nerve has to tell those muscles, hey, it's time to contract. This is self-stimulating. It does it itself. No nerve needed. It automatically contracts. If you remember back to when we were talking about cells, we started talking a little bit about cardiac muscle cells. And we said that if this is one cardiac muscle cell, and this is the next one, they are connected up by what's called a communicating junction, a gap junction. And it had a special name. Anybody remember what the name for that junction between two adjacent cardiac muscle cells is that lets them communicate with each other? It's a word you need to know. It's called an intercalated disc. It's a type of gap junction. It's really important that your cardiac muscle cells can communicate with each other. Because if they're not communicating when they beat, how are they going to beat? Out of rhythm. You need your heart to beat as one unit. Those cells all have to contract at the same time. So these communicating junctions, these gap junctions, let them do that. Take a sidebar for a minute. If these cells were plant cells instead of animal cells, what would we call that junction that let them communicate with each other? Plasmodesmata, right? That's a word everybody always likes and stuff. Make sure you know what that is. Last year, they were taking the AP exam, and Brett Melody, plasmodesmata was his favorite word. In about every class, he said something about plasmodesmata. Like question 5 on last year's AP exam was, what's the name for the communicating junction between adjacent plant cells? It's plasmodesmata, and everybody about the same time got to number 5, and the room just broke out in laughter. Because everybody in the room knew what it was because of Brett. So know what plasmodesmata are. Anyway, inside the heart, well, outside the heart, first of all, it's surrounded by a membrane called the pericardium. What's peri mean? Like perimeter means what? What's the perimeter? It's what surrounds something, right? So it's surrounded, the heart is. Cardium means heart. It's surrounded by a membrane. But let's get more inside the heart for these last few minutes. Inside the heart of a mammal, we're talking about human hearts right now. We'll get to some other things later. Inside a mammal heart, there are 4 chambers. 2 on top, 2 on bottom. The 2 on top are usually smaller, not as muscular. They're called atria. That's the plural. What's the singular for one atria? Atrium. Okay. Whoops. I don't know what the deal is with that today. Try that again. So it's atrium is the singular. All right, and then on the bottom, we have 2 bigger chambers called ventricles. Important point. The atria are basically like receivers. They receive the blood coming back from the veins. That's their function. They just receive the blood. The ventricles are the actual what? They're the pumps. Okay? So the atria receive, the ventricles pump. Atria receive, ventricles pump. Remember, your heart, since it's 4 chambers, is actually how many pumps? It's actually 2. One side for what kind of blood? Oxygenated, the other for deoxygenated. Which side is which? Which is the oxygenated side of your heart? The left. The left pumps oxygenated blood to all your body. So it's the big, strong chamber because it's got to pump the blood a long way, head to toe. The right side is much, the wall of that right ventricle is much thinner. It's not nearly as strong because the right side is simply pumping deoxygenated blood from the heart, right here, to where? To the lungs, right here. So not very far. So it's kind of weak. Doesn't need to be strong. If it was too strong, what would happen at the lungs? You start blowing blood vessels, right? That wouldn't be good. It'd be too much pressure. Let me show you a picture here. Here's the inside version of the heart, right here. Now remember, the heart would be sitting like this. So left, my right. First of all, there's a wall that separates the right and the left. Completely separates. That wall is called a septum. Where else in your body do you have a septum? Your nose, right? Separates your nostrils, the nasal septum. Septum is a wall that separates right and left. Now, it's hard to see on this diagram, but notice how the wall here is thicker than the wall over here. Because this is the side, the left side, that has to pump the blood to the whole body. This side, only pumping it to the lungs, not very far. Let's look at our chambers while we're here. Here's the left atrium. Notice it's small. We said it receives blood. Left ventricle, bigger, stronger, thicker. Here's our right ventricle. Again, pretty big, but not very thick, not too strong. And here's the right atrium. Again, it's receiving blood. Notice between those chambers, there's like a doorway and a set of valves. Between the right atrium and the right ventricle, there's a valve called the tricuspid valve. Why is it called tricuspid? It's made of 3 flaps. Cusp for like flaps. 3 flaps. When the blood goes from the atrium to the ventricle, do you want it to be able to go backward? That's what that valve's about. Keep the blood from going backward. Same function they had in the veins. In the heart, valve do the same thing. They keep blood going one way. And don't let it go backward. Over between the left and the left atrium and left ventricle, there's a valve there too, and it's called the bicuspid valve. Or sometimes you'll also see this valve called the mitral valve. And I'm going to bet somebody in this room probably has a problem with that valve. What do we call it when you have a problem with that valve? A heart murmur. It's a little leaky. You have it? It's usually not very serious. But when a doctor's listening to your heart, they can hear it, kind of like a drippy faucet. Some of the blood that should be going from here into the aorta leaks backward, and they can hear that. That's a heart murmur. Like I said, typically not too bad unless it's a really serious leak, but it can be bad. Let's look at the vessels now. Actually, let's go over here. The one thing that you got to know to pass this heart test when we have it is you've got to know the order of the way blood flows through the heart. We'll end with this today. You've got to be able to put these things in order. Now remember, it's a circle, so it doesn't matter where you start, right? But you got to know the order. It's a loop. So let's go back to the picture, and I'll walk you through the picture with this stuff. Let's say you're a little blood vessel, or a little blood cell, red blood cell, and you start right here in the left ventricle. So you're in the left ventricle. That's where we're going to start. From the left ventricle, the blood is first going to flow through this valve. What do we say that valve's called? Bicuspid or mitral valve. So left atrium, through the bicuspid valve, into this big chamber, which is the left ventricle. From there, the blood is going to get pumped out through this valve, which is hard to see, maybe, in this picture. That valve leads into the aorta. It's called the aortic valve. So from the left ventricle through the aortic valve into the aorta. It doesn't, it's behind this. It doesn't actually go cut through there, it's behind. So left atrium, bicuspid valve, left ventricle, through the aortic valve into the aorta. Where do we say the aorta goes? Throughout the whole body. All the arteries branch off of it. So after the aorta, I want you to put it through the body. So aorta to the body. In the body, that's where all the good stuff happens, nutrient gas exchange. When it's done, the blood starts to merge back together into what? Venules and veins. And eventually all the veins merge to form these two guys, superior and inferior vena cava. So that's what I want you to put next. Body to superior and inferior vena cava. Those carry the blood back to heart, but specifically, what part of the heart? The right atrium. So the right side of the blood is about receiving and pumping what kind of blood? Deoxygenated. That's why I use the blue. So superior and inferior vena cava to the right atrium. Then when these contract, the atria through the tricuspid valve into the right ventricle. From the right ventricle, blood is going to pump this way, through this little valve right here. That's called the pulmonary valve. What do we say pulmonary means? Lungs. It's headed toward the lungs. From the pulmonary valve, it goes into the pulmonary artery. Pulmonary artery takes it to the lungs. So, right ventricle, pulmonary valve, pulmonary artery to the lungs. At the lungs, it gets oxygenated. I'm just about finished. And it comes back to the heart from the lungs to these guys, pulmonary veins. Important thing before I finish here. It looks like these veins go into here. They don't. They all wrap. The pulmonary veins, all of them, carry blood back to the left atrium. So we just completed the loop, right? And that's where we stop. So remember, you could start anywhere. On the test, I might say start at the right ventricle. Go in order. Start anywhere. We started at the left ventricle. No, we started at the left atrium. Left atrium. Yep. Yes, sir. Yes, ma'am.
I wrote down 384 to 391. I just started during 3 o'clock today.
Yeah, you should
I
have
think
391.
I thought
for you guys, that's what I saw. That was for the other class. Yeah.
I put that on my
Yeah.
phone.
No, you guys are good. You guys are good.
I told other people it's not. I
No,
know.
no, no. For your class, that's what I assigned.
Okay.
Okay. Just make sure you bring it back. Mm-hmm.
There was something that works, it was strings. I didn't know, so I drank it anyway. If I died for 2 days, we can blame it on that.
We'll have a memorial service.
Like on my so it's still gonna say die second time.
Yes. Huntsville High School.
You guys have a great time, everybody! Hey, if you guys can tell me what was in this cup that I wasn't supposed to drink that I did, gratefully appreciate it. I drank it. You're like, watch the cup. I'm like, what?
Probably a lot of bacteria.
Yeah. Just makes you manly. Makes you manly. We should have had him in here. He would make things a lot more lively.
Start like making remarks about everything.
Yeah, it would be fun.
Dr. Dean,
Yes.
somebody smeared my drawing.
Sorry. I didn't do it. I'm innocent. Maybe.
Sure.
Does it say is your back hurting?
No, it's just.
Oh, so you carry your monkey, but come back. Gotcha. How is that in the hallway?
Yeah.
Oh, okay.
It's alright, keep biology exam.
May 14th.
After May 14th, we ain't doing nothing. Except